Translation in prokaryotes. Protein synthesis. It is the decoding of messenger RNA and converting it to protein. So here we have our ribosome. It is the location, it is the main spot where we see protein synthesis taking place. So we have our ribosome. Now, since we are talking about translation in prokaryotes, this ribosome is 70S ribosome. So it's 50 here up top and then 30 here on the bottom. Ribosome, 70S ribosome. Here, this multicolored chain is our messenger RNA. Now, this is multicolored to help you realize that there are codons within the messenger RNA. Now, a codon is a three set of nucleotides that code for amino acids. These codons are called sense codons. So I'll give you the first example here. In green, we have a codon right here, and it codes for, well, it's AUG. That's the codon, and it is our start codon. Our sense codon. So we will see our transfer RNA come to this particular codon because on the bottom of this tRNA is the anti-codon. It is DON. It is the complementary to this codon. So it links up and then it has, or it connects, it binds to that codon, and then it has an amino acid attached to itself. And then it'll go through the process, through the ribosome, and then the amino acid is left. Then we have our next codon. Our next codon, that is a set of 3 nucleotides that code for a specific amino acid. Then we have our amino acid and our anticodon, and I'll just abbreviate for AC, anticodon attached to it. And then what's left is the amino acid. And then this process goes on until it stops. So we have our amino acid, our anticodon that codes, that is complementary to the codon on our messenger RNA. And then our amino acid is left. And then this will go on until, like I said, it reaches the end. And the end will be a set of nucleotides. It will be a codon called stop codon, or nonsense codon. Now there are 3 of them, and I'll go over those 3 right here. So our stop codons here, this is how I would remember it. So it's 1 UAA, 2 UAG, and 3 UGA. So going across, we have UAA. Going down, starting at the U, we have U. Now going from top diagonal going down, it's U, A, and A. All right, now we have 2 blank spots, and we're going to fill those blank spots with G. And that's how we remember our 3 stop codons. All right. So once we have all of our amino acids, and I'll go ahead and do that real quick. Okay, now we have all of our amino acids. They have to bind to each other, and they do that through or by a peptide bond. So we have a peptide bond. And this whole protein is called a polypeptide chain. Oops. And this is translation in prokaryotes. Need more help? Click the link in my description box to get started with your microbiology study module.